Hello, everyone. Thank you for joining us today. Welcome to the Data Science Seminar from the University of Washington Science Institute. My name is Nicoletta Cristia, and together with Dr. David Beck, we are co-hosting the Data Science Seminar series. This is the first seminar of the quarter, and we will remain on Zoom for the entire quarter. We will start our seminars with a mini series of talks featuring projects from our Winter Data Science Incubator program. Today, we will have Will Kumler, a graduate student from the School of Oceanography, and our own Bernease Herman, data scientist at the Science Institute. Before we begin these presentations, I would like to invite our colleague, Dr. Brina Hazelton, the director of the Incubator program, to briefly introduce the eScience Winter Incubator project. Brina, I would like to turn this over to you now to talk a little bit about our programs.
Sure, thank you, Nicoletta. So the eScience Institute runs this incubator program in the winter time, and it's a quarter-long intensive collaboration between somebody we call a project lead, who the people who are going to be talking to you in this series, who can come from anywhere within the university, and one of our data scientists or research scientists. And the goal is really to address problems that can really only be done together. That, there's some data science aspect that the project lead can't quite figure out how to do, and it's in a domain where the data scientist or the research scientist wouldn't be able to do anything without working with the project lead. So it's really a collaborative project, and we run these every year, and we would love to see applications for anybody from this audience. I think we've actually gotten some people before this way, so this is a good way to advertise. It's quite a lightweight application. If you think you'd be interested in it, please come to one of the data scientists' office hours ahead of time, and we'll be happy to help you figure out how to navigate it. So as I said, it's a quarter-long series, so it's basically 2 days a week for a quarter that we work on these projects, and it's there's a wide diversity of projects that we work on, and you'll be seeing 6 projects over the next 3 weeks. So I'll leave it there. Thanks.
Thank you, Brina. Before we start, I'd like to make a land acknowledgement. As part of the University of Washington, we acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. Because we do have 2 different talks today, I would like to invite Will to start us off today. And after his presentation, you can ask questions using the Q&A button at the bottom of the screen, and Will will address the questions. And with that, I would like Will to start his presentation.
Fantastic. Thank you, Nicoletta, and thank you, Brina, for introducing the program as a whole. Yes, as mentioned, I'm one of the project leads for the Winter Incubator. I'm a 4th-year grad student in oceanography, working with Anitra Ingalls, and have been doing a good amount of research in the lab where we focus on mass spectrometry and particularly characterizing molecules from the ocean and characterizing the marine environment through this modern and very interesting particular bit of technology. But I actually started out and came to Seattle 4 years ago, as I mentioned, and one of the first things I noticed was if you were ever lacking for a conversational topic, you could always ask people for recommendations for hikes or for mountains or for what peaks we should go and find. And so having been here for a little while now, I have decided to compile my own idea of what peaks are best. And this is one that I've stolen from the Seattle Met, and I think it does a good job of visualizing some of maybe all of your favorite peaks. But I'd like to go ahead and share with you today what some of mine look like. These are peaks. These are chromatographic peaks actually from the data and from the projects that I'm working on. And each of these are very interesting because they can represent a molecule in the samples that we run on our liquid chromatography and mass spectrometer. If you're unfamiliar with this particular type of data analysis, it's relatively simple. What we take is we actually will run samples that we take from the ocean. We'll do some extractions and do some processing on them, and then we put them onto this beginning of the high performance liquid chromatography column. This column will then separate them by their polarity, how well they stick to the column. And so it actually lets you identify that there are multiple molecules coming out over time. And the instrument then measures their intensity. As you can imagine, liquid chromatography alone has the issue of overlapping many of these peaks and making them hard to separate. So this is usually combined with a mass spectrometer that adds an additional dimension of this mass to charge ratio, where we're able to separate things based on their atomic weight. This combination makes it very powerful for identifying different compounds, some of which are known, some of which we can identify a priori. We know that they should be in these samples, things like guanine, the nucleobases that are making up DNA in all of our cells, as well as more interesting compounds and different compounds in the ocean that have different purposes. I've highlighted 2 here from this particular chromatogram. But of particular interest to me are also the unknowns. And so these unknowns can only really be detected using some data-driven techniques. It's easy and simple enough to know if you have an idea of a molecule that's out there to look up its atomic mass on PubChem or the internet anywhere. And then you can go and look in your data at that mass and see whether you have one of these peaks that corresponds to the molecule itself. But these unknown peaks, you don't know their mass of to begin with. And so you have to use this kind of new algorithmic way of going and finding them in the data. And so this becomes a very challenging peak picking and very complicated algorithm style finding their way through these data, these sometimes massive amounts of data, up to terabytes or hundreds of gigabytes in size. So these unknowns are very interesting to me, and we end up using them we end up with some struggles because there are many of these unknowns, and oftentimes these peak picking algorithms are not very perfect. They're not performing very well. So here's a subsample of the few peaks that the raw output from the peak picker has produced, and it has decided that all of these look like very good, very excellent mountains. And I think many of us in Seattle might disagree with those, having been spoiled by seeing Rainier or the Cascades each morning. But our goal here, and the project that I pitched, the incubator in particular, was trying to organize these in some way or figure out which ones of these are the best looking peaks, because the peaks that you can see most of here are actually just noise. The mass spectrometer is picking up on some kind of background signal the entire time, and it's not corresponding to a nicely integratable, nicely observable particular compound or a particular peak corresponding to that compound. And so our goal in this project was really to go through and find some way of separating these peaks out by quality, by how nicely shaped they are, by how likely it is that a real mass spectrometrist, if they went through and looked at all of them individually, would assign them a real or useful data value. So then we can go through and filter out just the very best ones, leaving us with just a high quality data set without needing to go through and look at every single peak manually. That's kind of the goal, especially as the data sets get larger, it gets more and more manually intensive and really doesn't scale. This is an active problem. Again, I've shown a few of these, but you often have these issue across multiple files as well. So I showed just a single file in that previous one, a single sample, but you can imagine as you add additional samples, this becomes even more difficult. Going through each of these peaks individually, you can already see that this would be a huge amount of effort to go through and look at each single one of these traces one at a time, deciding whether it's good or not. And so there's this clear demand for doing this quickly, rapidly, and improving both the algorithms themselves, as well as what I focused on, assessing the quality of the algorithms. So I started out by going through and labeling all of those things, the exact thing that we said we didn't want to do, but we will need to use a data set, and many of these don't exist in the wild yet, especially the ones that we're interested in where they're looking across these many different files simultaneously. And the question is, why are there so many bad peaks to begin with? Surely the algorithms themselves aren't actually that terrible, and this is a very intentional decision on the behalf of the algorithm designers. In particular, we're seeing up to 80 or 90% of these as bad peaks. When I went through and labeled my entire data set, I found about 250 good ones and about 1,600 bad peaks that I wouldn't have wanted to include in my downstream analysis. And this is because the peak picking algorithms are designed to minimize the number of false negatives. They're designed to make sure that you're not missing out on any interesting signals. They're arguing that you can always do some downstream filtering, you can always go through and sort things out later, but we don't want you to miss anything from the very beginning. The question is, if we accept that we're going to miss out on some signal here, rather than introducing all of these essentially false positives, these peaks that are bad but are being labeled or identified as potentially good, we're willing to accept that we're going to miss out on some of those. Can we massively speed up this process by mitigating the number of peaks that we actually have to then go through and look at? This is a very common machine learning algorithm, or very common machine learning motif, where we're training up then a classifier. We're trying to find things that will classify these peaks when we hand it to them as good or bad. And this is kind of the overarching goal of the incubator that we were working on. We tried a couple different options that I'll talk about in very brief detail. But the information, and the reason that we think this might actually work or perform better overall, is that we can include information not just from the single individual trace, but from multiple samples as one big example of something that we think might improve, rather than going through and doing the peak picker and just using the peak picker output itself. So when I went through and actually did these, we sat down and looked at a bunch of peaks from the very beginning. We tried to start deciding on what distinguishes these good peaks from the bad ones. What are we noticing when we say that looks like a good chromatographic peak versus that looks like it's noise? So this is the very classic feature extraction in machine learning or AI algorithms, where you sit down and you try and calculate some metrics that will separate them out. Some ones that are commonly used in the literature or very commonly used as kind of thresholds that exist currently are something like a large area. If it's a bigger peak, it's more likely to correspond to an actual compound is kind of the thought process, as well as these other ideas of signal to noise. This actually doesn't work as well as we'd like it to. You can see that the bad and the good peaks are kind of stacked over each other. And indeed, when we run a very simple logistic regression on this one, we never even get above 50% good peaks, no matter what the size of the peak is, even though you can see it kind of tailing up at the end there. So it's going to be improving. The larger the peak is, there's a little bit of truth to this intuition. But we can do better. So with some of the custom metrics that I came up with and that we worked on in the incubator, we were coming up with some additional ones for peak shape, how Gaussian it is, how much it actually looks like one of those mountains we might see out like Rainier, as well as things like, is there a difference between different treatments using some biological information? So I'll talk about the different strengths of these metrics later on. But this one you can see actually is performing a little bit better because we're getting separation between the bad peaks colored in red here and the good peaks colored in gold. And here a very simple logistic algorithm will do quite well. We ended up working with a multiple logistic model. We did try some fancier ones with some classic clustering, as well as the random forest classifiers. And we found they're performing very similarly, and the logistic regression was very easy to explain and very much a powerful linear model that we ended up using. So from the initial model, I was going to go ahead and share what this looks like. And what we found first was that the very bimodal is very confident. It's either saying like, yeah, most of the time I'm very sure this is a peak or very sure it's not a peak, which is already very encouraging that it's able to do something. And it's not just bimodal, but it's actually doing a good job of separating that good and bad distinction. If we zoom in on just the bottom portion of that peak, just to give you some sense of what the variation actually looks like there are very few peaks along the very bottom, sometimes just 2 or 3 at a particular area. And so the specific threshold here is not very critical, which is very reassuring to us. Sometimes deciding on a threshold can be a very difficult or very challenging idea. But here it's actually quite flexible as far as what threshold you need. I've rebinned them to show this a little bit in more detail, where I'm using much wider thresholds with 50% in the middle and then stepping outwards by essentially an order of magnitude or 2 each time to show that if you were interested in it, you could actually identify very, very high quality peaks and be very confident those are very good, or step much lower and identify just the very worst peaks if you really did want to get every bit of data, every bit of possibly good peak out of this data while still removing up to 600 or 700 bad peaks. So this is where we talk about, again, that false positive or negative trade-off. When we started out with the model, all of these peaks have initially been flagged as essentially good. You could put these into a downstream analysis, but it wouldn't be particularly useful. And this is mostly essentially erroneously flagged as good peaks because we don't want them to look the way that they do. And so we're instead just trying to move things from this false positive column into that true negative column is our goal. We're trying to minimize these 2 columns in the middle where we get false negatives, where we think that something is bad and it's actually good, or we're trying to minimize the false positives where we think something is good and it's actually bad. So when we actually did the very basic logistic classifier, again, this is kind of the kitchen sink. We threw all of our features into the model and we tried to get as good a separations. We didn't actually perform quite well. This was very reassuring to us to see that we can in fact move most of those features from this false positive column into the true negative because those peaks were in fact bad. Notice that however, we have actually added this column of the false negatives where we're missing out on about 13% of all the peaks that we flagged as potentially good. So we are accepting that compromise using this particular threshold here. And the trade-off is that we've then gotten a very low rate of those things that are bad that were flagged as good in that true positive, in the false positive column. But that's not very interesting because that was all just done on our own data, on the original training up data. And what we're interested in is how well it performs on some new data. If we handed it a new set of data, how well it will perform. This is actually the initial result, is that we got a similar bimodal distribution. Again, this classifier is very confident that things are either very likely to be a peak or very unlikely to be a peak. And it's not actually as accurate as it was before. So we're expecting some kind of degradation, and this is a major concern for us, is how much it degrades when applied to a new data set, whether we can continue to keep those low thresholds of false positives and false negatives, or whether the model is going to decay and actually introduce a whole lot more. So again, I went through and labeled this entire second data set, so we have this kind of way of comparing the two, and we did keep them relatively low. We see some inflation, particularly the false discovery rate, where we're getting a lot more peaks that the model thinks are good that I thought were bad. But overall, it's not enormously increasing, and instead we're still getting nearly 1,000 peaks that we no longer have to even look at. We no longer even have to consider whether or not they're good or bad when we're sorting through these, which is enormously helpful to begin with. So one thing that we can also do now that I've labeled a second data set is actually reverse this question. I'm working with two data sets here, one of which is the MESO data set, one of which is the Falcor data set, named after the larger projects they're coming from and the oceanography that is actually driving these questions. And so we can switch these two data sets where I've been using the Mesoscope data set as the training and the Falcor as the testing, but we can also reverse those and include the Falcor to see whether this is a reversible model, see what the performance looks like. And these results are quite similar to the ones that we had before. Again, there's a slightly inflated error rate when we switch across data sets, looking at those plots on the two middle columns on the right plot here, where it's slightly higher than it was on the left side, but this is about what we expected and it's relatively consistent. And again, still removing an enormous number of peaks there. Here we've removed 1,500 peaks from consideration, and we've missed out on just 55 potentially interesting compounds. And you could change that with the thresholding as well. Now, having shown that this model works, one of our questions was, do we really need the entire thing? We've got nearly 25 features in here, and so I'm going to show you the same plot again just to bring that back. And this has really all of our parameters in it. This says this is all 25 of those, and we're not sure which of those is the strongest, which ones we actually need. There's a lot of redundancy there. We did sit down initially and go through and remove those things that are most strongly correlated, which can be an issue for these logistic regressions. And so the things are mostly uncorrelated at this point, and mostly we attempted to make them more or less Gaussian with some transformations as well. But these are still all of our parameters, and we really think that we didn't need all of them. So one obvious option is to just take only those default parameters that come directly out of the peak picker. There's a lot of information that comes from those peaks directly that is not immediately interpretable, but can still be used to try and predict good versus bad peaks. So either this model performs a little bit worse than the previous one. Again, those columns in the middle is what I'm keeping track of. And they're slightly higher here than they were before. And they especially seem to be increasing a lot as you move onto the different data sets where those are severely inflated over there. And then I was curious about how these custom metrics that I'd come up with would perform. And so that's what I'm calling the raw data parameters, because you actually do have to go back to the original raw data, that original peak that I showed you, and recalculate things from now. This isn't something the peak picker reports automatically instead. So here we see actually a slightly higher or a reduced performance on the initial model. So this left side, the same data set, is actually a little bit lower, but it doesn't seem like it increases so much. And this is something I sent to Brenna when we were discussing this, and I was like, is this confirmation bias, or is there actually not as much of a trend here? This seems to be a little bit flatter, which would be reassuring, because when we're looking at different data sets, that might inflate it a little bit less. So Brenna thought maybe perhaps the same thing, and so I'm going to go ahead and show what that looks like. Again, I'm just pulling out those middle columns here and highlighting two metrics of which we're particularly interested in. I've called them the number of good peaks found, where the higher number is better. And so this is the question of all the good peaks that I could have identified in this data set, what % of them did we actually get? This is very similar to the recall that you might be familiar with if you're doing a lot of training, as well as the false discovery rate. Again, of those things that we decide or the classifier thinks are good, what % of them are actually bad is another question. That would be something like 1 minus the precision, if you're, again, familiar with the terminology used here. So I'm starting with, again, all the different parameters. These are the 3 different models that we were testing as far as some different things that are useful. And I'm going to go ahead and show the ones on the left to say, yes, we're getting, again, most of the time, a pretty reasonable good peaks found percentage, as well as a reasonably low false discovery rate that does decay a little bit downwards in the first case, downwards in the good peaks found, and upwards in the false discovery rate. The model as a whole isn't performing quite as well as it did on its initial data set. With the defaults only, it seems like it's a little bit more aggressive. And then with the raw data, maybe a little bit flatter. I've eyeballed some lines here to show that. I wasn't really comfortable doing some actual fits, because this is what we're interested in projecting to, is a data set that's even more different from our training data. We're interested in asking, like, okay, so if I threw a different data set that's wildly different in here, how much would the model degrade, or how much do we have any control or any sense what these 2 important metrics would be? And the concern is that we've, again, managed to overfit our model with all of these different features that we're using here. And so the lines that I've, again, eyeballed, haven't done any actual fitting here. These were added in PowerPoint about 4 hours ago. I've gone ahead and eyeballed where I think they might go. And again, we had a data set of, again, increasing difference from the training data. And this is, again, arguing that we should use more of these raw data metrics, even though they do require going back to the raw data and require some additional time to calculate them. They seem to be persistently a little bit more robust across data sets, even though their initial performance is relatively low. So this is a very difficult question for us, is deciding when the data set is different or how different it is that I'm interested in exploring in the future. But it was something that we were looking at to say, like, hey, maybe we only need these raw data metrics. And I can point out as well that these raw data metrics actually just consist of 2. So the full model, the full thing with all the parameters says about 25. The defaults conclude something like 15 to 20 different parameters in there. And then the raw data is actually just 2 metrics, one of which I showed you earlier with the peak shape that did a very good job of separating those out. And another one is a custom signal to noise estimation that was, again, has been talked about in the literature. So this is kind of where I'm imagining these data sets might end up, but I'm flagging them with a question mark here just because I really don't know. It's hard to say how different a data set is from the previous ones or how similar. And so this is kind of an uncontrolled unknown that we'd like to have a little bit better sense of where it might end up. We also want to say that the ideal model would be independent of the training data, right? In theory, if you have a really robust model, it doesn't matter whether you trained it on Falcor or on Miso. You could get the same data, you could get the same model and the same predictions, whether it's trained on one data set or the other. And so this is something that we tested as well. I'm going to show you what this looks like with the full model, all 25 of those parameters initially, comparing the estimations, the likelihood that this is a real peak from the Miso trained model to those from the Falcor trained model. And so again, we're expecting this to be a line, right? If one model thinks that a peak is good, it should also be good in the other model. If it's not, then it really should be low in both of these. What we end up with is a pretty good scatter throughout here where there's a lot of disagreements, where a lot of the peaks that were trained up by the Miso model actually end up falling quite low on the Falcor trained model. Again, the overplotting is hard to show here, but do remember this distribution is very bimodal. So we have a lot of peaks in that top right corner and a lot of peaks in the lower left corner. But the interesting thing is to compare it then to one where it's just trained up on those 2 metrics of the raw data. And this is telling us that we've got a much closer fit to that 1-to-1 line. And we think that we've done a much better job of not overfitting the model. Again, initial performance is a little bit worse, but we haven't overfit the model, and so we can be a little bit more confident moving on. Even more importantly, we'd like to think about this in ranked space as well, to think about users. So you might start out and say, okay, show me the best peaks in this data set, and I'll step through them. Show me the very best one first. I'll decide whether that's a good or a bad peak and step downwards. And so we really want to make sure that the rankings are the same. So if you start out with the very best peaks, do the models agree on that one? Does it really matter what your training set was for these particular questions? And this is where we get a very nice correlation using just a raw data matrix, where the full model tends to have a lot more spread in there. Some pretty severe disagreement about what actually are the best or highest quality peaks. Again, reassuring us that we haven't overfit the model when we just use those 2 parameters, but we might be doing so when we use it. So that's the step down training that I was just talking about, where I can imagine a user interacting with this one and labeling until they're essentially bored or they realize there's no real recoverable data left because they've seen so many noise peaks. In the future, I'd like to continue looking at these even more different predictions. We're working on some additional data sets that are not very similar to these ones, but does require more labeling from me or from some other mass spectrometry expert to go through and identify these peaks as good or bad. One way we're hoping to make this a little bit easier is to go through and identify just the good ones and see what our false discovery rate is, but it's much harder to get a sense for what the false negative rate is or how many peaks we missed out on without labeling the entire data set, which we're hoping to avoid. I'd like to turn this into a package, so again, make it a little more helpful for other people. It's currently, a lot of it is up on the GitHub, but is as scattered scripts, and there's no unit testing or any kind of control over things. And so I'd like this to accept the input from the peak pickers. There's a couple different algorithms that can be used, and not all of the features can go into each algorithm. And so what I'd be interested in is creating a package that uses the features that come out of these different algorithms and predict essentially the quality or the likelihood that each peak is a good one directly. And we can always do additional feature engineering. If you have great ideas for what you think might separate out a mountain-looking peak from a noise-looking peak, I would love to hear them. I love thinking about these ones, and I'd love to put it to the test to compare with the features that we already have. Again, I'm happy to add additional features and continue talking about them. So with that, I'd like to acknowledge the whole crew that did this. There's Brenna Hazelton, my eScience liaison, who's been absolutely fantastic in answering things, whether I send them to her at 11 p.m. on Slack or show up in her office bright and early in the morning and have some bright ideas or some terrible ideas about how things are going. The entire eScience team was a huge resource the entire time. Everyone on the 6th floor is absolutely fantastic and super friendly, whether I've got a one-off question or a very detailed one that I'll need a lot of help with. The Ingalls Lab shown here, both present and past, helped collect the data, helped teach me things that I've been working on, and have been a huge support throughout the process, as well as my funding from the SCOPE program and the Simons Foundation. With that, I'm happy to go ahead and take any questions. I see that we've got at least one in Q&A, but feel free to drop additional questions in there and I'll do my best to answer them. But thank you very much. I'll pass it back to Nicoletta.
Yes, thank you, Will. That was great. An excellent example of feature extraction. And I think maybe we do have time for one question that reads, what are the algorithms other than logistic regression? I think you did mention something about random forest. Could you elaborate a little bit on model selection?
Absolutely. It's fun to be chatting with a group of folks who are very aware of these different algorithms and interested in how they performed on this data. When we started out, we were hoping to find just some kind of clustering algorithm would identify these. Again, visually, they seem very distinct, those good-looking peaks versus these just completely noisy peaks. So we were hoping that some kind of clustering would identify these are the good area, these are the bad area. And I've had some success using some raw data, but those aren't quite formalized or finished up yet. The other one we did try was the random forest. The random forest has some incentives as well, namely being able to not have a linear response. You can have things like above a certain threshold and below a certain threshold. So you're able to identify those things that are only good in the middle. That works really well. And that was really kind of where we left it, because once we started seeing that the logistic regression and the random forest were performing about the same, and a big emphasis of ours was on interpretability here in this project, where there are algorithms out there where you drop it all into this massive neural net and then you get some outputs, and it's really hard to tell how well it's performing or how well it might perform in your data set because you don't always have access to the training data either. That was the main algorithm that we ended up settling on, was the very simple, very intuitive, and very explanatory logistic regression. But we did try random forest and clustering as well.
Excellent. There are a couple of more questions. One, I think, refers to your future work, and just reads after, what is after you find the peaks? Like, I'm assuming it refers to, like, what do you do with the data set that you just generated?
Yeah, well, you turn them into beautiful graphics and add them to a presentation, of course. No, the real goal here is identifying interesting molecules in the environment, right? So I've mentioned that these samples were taken from the ocean, and we've processed them. And downstream, what we're doing is we're looking for interesting biomarkers of individual species. We're looking at how these molecules change in concentration and abundance across different environments in the ocean, change with the seasons or over the days. Oftentimes, if we get high quality data, we can also talk about the exact concentration of these molecules, starting to reconstruct the composition of seawater or the organisms that live in seawater with high accuracy. So eventually it does turn back into biology. It turns into the chemistry part where you're figuring out what the molecules are, and then it turns into biology where you're figuring out what they're doing. And this is the very first step along that process. And that's why I'm focusing on it right now, is because it does affect all of those downstream analyses.
We got a lot more questions, but I don't think we have time to address them all. So please contact Will. He will provide his email address. Do you have it?
I didn't add it to this slide, but I'd recommend folks actually look at the Incubator website. My email address is up there, as well as just a quick summary of the different projects that you'll see. I am happy to answer questions by email, and I'm happy to go over the figures with you again if you're curious about those too.
Okay, let's take one. Maybe.
Yeah, I was going to say, just as the next person, I think I have 5 minutes until I was technically supposed to start, and I'm even happy to go beyond that.
Okay, Bernice, keep going.
Bernice is throwing me under the bus. She wants to hear more about the.
Yeah, I want to hear more. Less me, more Will. It's always great.
I'll start with the last one. Is it possible to get a better description of the evaluation parameters? You mentioned a measure of peak shape and one other measure from the raw data.
Yeah, so thinking about the features that we extracted and what we did to engineer those. Yes, so the two metrics that ended up being most useful and most interesting were calculated by fitting an idealized kind of curve. So these peaks that they come off the instrument are very typically Gaussian, and the very good peaks are highly Gaussian, maybe with a small tail on one side or the other. And so what the two features that we did best with was, or were calculated by, was fitting an idealized peak to them, where it was zero noise at all, just a perfect Gaussian with a small degree of skew. And then we would look at the correlation between that perfect idealized peak and the one that the real data looked like. And so you can imagine if it is also a very clean, very Gaussian peak, it's going to have a very low it's going to have a very high correlation with that idealized peak. And so that's actually the metric you may have seen, that it went from -1 up to 1, and our very best peaks were piling up against that very perfect correlation, the 1s and the 0.99s, while the noise peaks tended to fall closer to that 0 or the 0.2 kind of area where we'd expect kind of random noise to fall on that correlation matrix. The other metric that turned out well was this metric of signal to noise. There's a lot of different algorithms out there for measuring signal to noise. The one that I ended up using that seemed to work quite well, we actually did compare both the signal to noise that came directly from the peak picker, directly from the algorithm that finds these peaks, to a custom one that I came up with. And the custom one actually did perform a little bit better. And that was calculated by taking, again, that idealized peak and subtracting it from the raw data, leaving just the residuals behind, and then calculating the standard deviation of those for some estimate of how much variance there is if there hadn't been a peak there in the first place. So those were the two there. A lot of the other metrics I actually don't have a good explanation for, particularly the ones that come directly out of the peak picker. There's very little information and very little documentation on what they do or what they're functioning on. And some of these algorithms that are peak pickers actually have zero documentation whatsoever. They're a complete black box where they're just provided as a Windows executable file, and you don't know how exactly they're calculating these. And so being able to go a little bit more open source and talk about, here's a simple algorithm, something simple that finds these, was, again, a major incentive in this project.
Yeah, I think there's another question related to that. Do you consider modeling peaks by standard mix LC chromatogram real peak shape? I'm going to answer a question that I think that might be asking, but please speak up if I haven't. We're thinking about, we have not only samples in oftentimes these runs, but we also have our standards, where we've taken the known molecules and added them to either water or some kind of representative sample or some kind of combined sample to capture some of the matrix effects that you'll get. And the standards are a tricky one because they're only going to show up in known files. I think that that would be a great way of actually training up an algorithm where you actually say, like, hey, there's only 100 peaks in here, but we tend to end up with standard mixes getting a lot more peaks that we actually don't know what they are. So the peaks there aren't necessarily a lot cleaner or a lot messier than the ones that we get from the real data. The ones that are from super salty, super low concentration ocean samples tend to be the most complicated, but really even just in pure water, we still get a lot of noise that's happening, and the peak pickers don't perform perfectly there either. So that's how I'm going to answer that particular question, maybe about the standard mixes as well. Thank you, Will. I think we'll stop here and turn it over to Bernice. Fantastic. Thank you so much. Thanks for having me. Bernice, are you able to share your screen?
Yes. Let me see here. I think I'm just going to go share the whole thing, and we'll then do this. Okay. Are you guys able to see the first slide?
Not yet.
Oh, no. It says I'm sharing. Okay. Let me try again. Let's actually, I'll just share on the other screen. Okay, how about now?
Now it's good.
Okay, great. I'm going to move my camera just a little bit so I'm not looking away from you. That will be not fun. But you know, I'll try to stay in the camera. Okay, great. Awesome. So thanks for having me. I am going to talk today about my project, investigating structure of social science research data sets for better ML evaluation. And a little bit about me. I am a data scientist at the eScience Institute since 2015. So I've been here a while, and I have a number of roles now across campus and elsewhere that one might be helpful for kind of my role in the incubator this quarter, but then also kind of my inspiration for the topic. So as a data scientist in the incubator, I have been a part of a number of incubator projects as well as data science for social good projects. Here's a couple of them here on the screen. As a data science liaison or data science lead to a kind of project lead pair, And this is the first year that I had a project of my own that I wanted to submit. So I had the interesting role of being both the project lead and the data scientist assigned to my project. So it was just me. But it was really great to do this work with the cohort overall. That was really great, and helped to have a lot of discussion and troubleshooting and kind of next steps that I wouldn't have had otherwise without the cohort. So thank you guys all so much for being a part of it, and having me there. I will say a little bit just about kind of the other things I do and what leads to the project and why I was interested in it. So there's a lot of different things that I think about kind of based on the different roles that I have, but some of the things that come together for this project are open source tools and tools that help researchers kind of with methodology. Is kind of a big inspiration for why I joined eScience and the work that I like to do there. Data summarization and evaluation is something that I think about a lot, both at the startup that I'm at Y Labs part-time, as well as research interests. And then finally, a SynRD project, which I'll talk a little bit about further, and the inspiration for using social science data sets. All right, so now what is the project and what motivated it? The motivations within the project are to investigate the differences in the properties and the correlational structure of data sets across two cohorts of data sets. So one being machine learning data sets that are very commonly used in the machine learning field, as well as some other discipline within science, starting with social science for this project. What I would like to do as well is find new ways to express correlational structure for tabular data. We'll talk about why that's a problem a little further in, but correlational structure is such a huge part of the advances that you see in machine learning for images and text and audio. And correlational structure for tabular data is quite a different beast and something that is a little less studied and thought about, or at least in areas that I've been. I'd love to hear people from other fields talk about how they think about correlational structure for tabular data. And then finally, build open tools to help researchers to better situate their empirical research within the broader realm of kind of possible data sets in their space. Okay, so what are some of the research questions that I was kind of going toward? Are there quantitative differences between data sets? We'll talk about how we're defining data sets that are commonly used in machine learning compared to social science research papers. Can we extend some of the existing literature and tools to include some correlational approaches, maybe something like graph-based data set metrics? And then for future work, how do these properties impact the efficacy of things that we build on top of those data sets, such as models, but as well as data generation techniques that I'm really interested in. All right, so this is an example actually from a paper that I am a co-author on, but it's pretty common on how machine learning researchers will compare multiple data sets when they're, for example, introducing a new data set benchmark, or even just in some papers, comparing the data sets that they do their empirical analysis on. So it's common in machine learning research now to have a number of data sets that you're doing this analysis on. And there's a number of metrics and things that we'd want to know about the data set. And so you can see things kind of like this. This is in particular is a hierarchical data set. And it's also like a multi shot or top K sort of model. So it's a little different, but things like the number of classes within the data set, the number of instances of data, the average amount per class or percentage per class, and then the 5 shots important when you're doing 5K. So these sort of metrics, this number of metrics is already a little more than you might typically see in a machine learning research paper about data sets. It kind of depends, but we're fairly limited, right? Like these things are just kind of thought of on our own of which of these metrics to include. And the overall goal that we're trying to express is largely that these two data sets are different in these ways or similar in these ways. But I'd love to think about how can we think about this a little more quantitatively. So what were my activities during the incubator to think about this? Well, the first step was to collect representative baskets of data sets from both machine learning and social science. We will only look at a subset of those today. So characterizing these data sets, so repurposing existing kind of metrics for data sets description and applying those to these data sets and comparing. Novel metrics, so things like exploring new graph-based metrics to capture this correlational structure issue that we'll talk about later, and then packaging, something similar to what Will said. What I hope to get to is to clean up the work that I've done and kind of share an open source package to make this data set comparison easy. And for something that authors of papers who are working with data sets can very easily kind of add some analysis to their papers to help kind of dive into the differences between the data sets that they're using to analyze their work. Okay, so the inspiration for the social science research and the social science research data sets themselves actually come from a collaboration that I have done with researchers at NYU, as well as 3 summer student researchers at ICU in Ukraine over the last summer. So I'm going to share a little bit about that project called the SynRD project before going to my own. So these are the collaborators, and it was really great working with all of them. And thanks for some of these slides that I'll use to describe SynRD. So for SynRD, it's a little bit more focused, this project's a little more focused on differential privacy. So actually the third research goal of looking at a type of data generator and evaluating it on new data sets. So we won't define differential privacy here, but feel free to remind yourself of that if you're familiar. But the research project for SynRD had 4 stages. So first was to find some social science papers with public data and high impact. The goal for SynRD is to first reproduce the original results. So that was a fun summer of us reading papers that we were not experts in and doing our best to reproduce the results. There's lots of things that we've learned about reproducibility in science and lots of difficulties. We had I think a very low perfect success reproduction rate, although we could reproduce to some extent a number of papers. For SynRD, the next goal is to run different synthesizers on that data, so differential privacy synthesizers, and then compare those initial insights based on that private data. And this, just to understand what data set I'm working with, we can look at the sorts of results that come out of SynRD. So this was an original figure for one of the papers. And this is a reproduced figure using our reproduction code. They are not often this similar, but you got to show the fun one. And then we would do some step like this, which is reproducing with one of the differential privacy algorithms. Okay, and then similar things for actual kind of tables and most importantly, findings of the paper, right? The analysis that they've done in the paper. Okay. So why is this important? This is important to know because this was half of my data set collection, was collecting the original results from the papers that we use from SynRD. So the whole SynRD process for the first step of this that I'm showing today wasn't necessary for this, but for future analysis, thinking about synthesizers and models that we build on top, it was important to use some data sets that we can replicate and rerun and do lots of cool things with. Okay, but where am I getting the data sets for my machine learning and social science respectively? I'm getting, for my initial subset that we'll talk about today, data sets from UCI, very popular repository of machine learning data sets, very old at this point, very common. And then for the social science papers, ICPSR, which is a research repository for social science data sets, and is very kind of well structured, lots of direct data downloads and metadata about the data sets that are used. Okay, and these are the baskets of data sets that we'll look at the results for today. So 5 different UCI data sets, and then 8 different social science data sets. Okay, so how do we characterize these data sets? Well, there is a number of metrics that you can kind of come up with. That table is the first example of that. But one resource that I found that I really liked is the meta features literature from the meta machine learning or meta learning field. So meta learning within machine learning is most popular for auto ML. And so this is sort of, can I come up with an algorithm or a model to predict the hyperparameters for a machine learning model given some new set of data. So it's like one step above. That's why it's meta learning, because not only are we training a model, but we're training a model to build models where we can choose either hyperparameters or sometimes architecture, neural architecture. But one thing that was really great that came out of this is this very empirical, kind of simple approach of saying, well, let's collect a number of metrics about data sets, these meta features. These are different properties about the data, and we can use these properties to, when we see a data set, predict the likelihood, so either use it to build a model to come up with these hyperparameters or predict likelihoods and predict performance. So I'm going to take these meta features and use them to do this analysis at a much larger scale than is typically done for machine learning more broadly. So the first thing that I did very fast, which was nice, was to populate a similar table to what we've seen before for this data set comparison using just a couple of these machine learning data sets compared to these 8 papers for the social science research. And already you start to see some interesting things come out of this, and we can calculate things like error bounds or confidence intervals for some of these metrics as well. Okay, but the really cool thing is, not just using these tables, but going beyond that and doing larger statistical analyses. So what I did is started with a just basic statistical test, 2 sample t-tests across all of the available metrics that I had for the meta features, and explored with a number of corrections for multiple hypotheses. So Bonferroni correction, Holmes correction, false discovery rate correction. And I didn't think, and I think I was correct to think that I wouldn't have time to dig into some of the actual features that come up, because they're quite frankly hard to explain. But there are some really interesting ones there in differences between my larger set of machine learning data sets and my set of social science data sets. And you can see the results of those when I link to the project. Okay, but let's jump over to novel metrics with the rest of my time. So all of these metrics are great, but we want to think a little bit about correlational structure. Okay, so this is an image, and this is something interesting about images and text to some extent, but images largely, is that while we call images and text unstructured data for kind of database reasons historically, in some ways there's a lot of structure there that we don't necessarily see in tabular data, which is the type of data that I work with most often. So in this example, there's a picture of a dog and a number of cherries, in closer view, labeled as cherries in ImageNet, oddly enough. If you use top 1, maybe that's why you should use multiple. But we can look here, I actually don't have labels, which is terrible, but there is, so if you look at the correlational structure, so if you're comparing pairwise 2 pixels left and right of each other, so side by side to one another, so for one pixel, if I look to the left of that pixel, and I plot this at the top in the scatter plot, we see that the color of that pixel is very similar, right? Like there is a strong linear pattern between these. And same for if we look at an image or a pixel that is one above and one to the right of each given pixel in the image. And so this maybe isn't surprising. What it tells us is that pixels that are next to each other have some sort of relationship, right? Like they are very similar. They're likely to fall along the same line as far as color intensity and grayscale in this example. This sort of correlational structure and these, so actually going back for a second, this sort of correlational structure is part of the basis of the inductive hypothesis, or sorry, the inductive biases that we have for our machine learning models when it comes to images and text and things like this. And so we have to kind of use these same sort of inductive biases for tabular data, but you run into a problem because this is an example of one of the tabular data sets that I used from UCI. And what you can see here is that we have features similar to images, but those features and their relationship to one another would be much harder to measure and kind of describe. So when I think about the correlational structure across features, it's not as clear. And we can certainly do things like this. So correlation matrix. This is on the left, one example for UCI Bank, and on the right, one example from one of the papers, to start to look into this correlational structure and describe this correlational structure. But another way to think about this is actually graphs. So converting tables into graphs for understanding the correlation between features. So there's a lot of work in literature that I dug through during this quarter to understand where the conversion of tables into graphs went. But it turns out that much of that work is for multi-table databases. So what can we do? What I ended up doing here is finding the target variable for each of these models or each of these data sets, and that becomes a node in a graph. And my graph picture is not showing up. That's upsetting. Is it a transition? No, it's not a transition. Okay. That's okay. So for example, we take our data set. Let's go back to here, UCI Bank. There is some variable that's most commonly used for some prediction tasks, let's say. And for each of these data sets, both for the papers and for the machine learning, I found that target variable, and those are the nodes of the graph. And those nodes have edges between the different variables related to the correlation, the shared correlation between them. And those are metrics that I'm hoping to do some further editing on and including into my set of metrics for this kind of automatic comparison across features. I'm really bummed about the graph. I will see if I can try to get it to show up somehow. But with that, how about I switch over to questions and end there.
Thank you very much, Bernice. I think we exceeded our time here, but I may ask you one question. You talked about social science data sets, and I was curious if your metrics vary as a function of what those studies were about. Like you did show some like cars and people and whatever, but I was wondering, what are the areas, social science areas that you investigated, and how this is going to impact your statistics and metrics?
Yeah. So first of all, I apologize. I thought this went till 5:30, but I think it's like the bell thing where we stopped 10 minutes earlier. So that's why I was like, oh, we're doing everything. The math is so weird. But okay, so I apologize for that. The answer to this is that the metrics stay the same. So I'm calculating the same metrics across all of them, but the specialization, I mean, really the feature engineering for these data sets have already been done. Partially, this changes as we kind of do some more automatic feature engineering in our data sets. But for all of these, the feature engineering is really the work that the social scientists did. And that's that work that we did to replicate all of their analysis that they wrote normally in text in the paper. So all of that feature engineering is kind of unique to each of the data sets. But the metrics that we calculate across them are shared. I calculate the same ones.
Okay.
Thanks. Yeah, sorry for running out of time. I miscalculated. I thought we had 4 minutes.
No problem. I think we can end our session here for today. Thank you both Will, Bernice, and also Brenna for joining us today. And I will see you next time. Thank you.